I think that's also a example that at the beginning we were trying to see if we can get a sequence from Siemens, but it took too long, I think. And we decided to turn to Pulseq, and I think it only took me like 3 months to generate a MRM, like a ISMRM abstract. So I
Good.
think
Okay.
that's a very nice example to show that the Pulseq is very important.
Yeah, and your sequence is really quite complex and has so many options. So I'm pretty sure you would spend more than a year in IDEA trying to implement all those possibilities and debug them basically.
And I
Yeah.
guess the
So
Friday doesn't have the Siemens clear case, I guess. Like they have some package called clear case where you basically have all the sequences, but I think the Freiburg doesn't have this
no, I
whole
don't know
set of.
what is clear case.
Okay. It's just like a package of all the Siemens source code. And usually a Siemens engineer will have that. And I heard some people from America also have something like that, but I'm not completely sure.
Okay. Well, I mean, we have very, very, very many source codes, but not all. So certainly not everything. And the things are that very often, you get the sequence source code, but what you need is in the kernel. API, like in case of the API, you have this API kernel and then API readout class. So two separate classes. And then sometimes you also want to know reordering. And so all these bits and pieces of the sources, they are really well hidden, and you request first source code and you find out that what you get is not enough. You ask once again and so on. So that's a long process.
Mhm. I see.
Okay. Well, I think we have many people online already. I think we can maybe start with some general announcements, right, John?
Yep, that sounds good. So the first thing is maybe settle on a date for the next meeting, so in March, and also we'll
Yeah,
probably, yeah, go ahead.
you asked me in the email, so I'm fine with the proposal. So we can certainly do that on March 3rd. March
Sixth, I
5th or
think.
6th. Yes, yeah.
Yeah, I think. Okay, so let's go with that. So then we'll try again just to ask for one contributor like we're doing today. It felt maybe a little too packed with two, and then we have a little more room to address any general technical issues, which is probably also useful. So again, you don't have to do it here, but you can contact me or Maxime if you have a project that you wanna talk about, get some feedback on. And as you know, we want this to be useful for you as well. So it doesn't have to be, you know, something that's already published. It can be something that you feel is ready, very close to ready to share, or you might have, a technical, a piece of advice or nugget that you want to share, or maybe an also unsolved problem that you've been thinking about and that you want to perhaps recruit the community to help out with. So anyway, we want to keep this really open. So just want to make sure that's the case. Um,
Yes. Or also, like you're also totally fine to speak up right now if you're really eager to present something. So just it's your chance now. Speak up or write in the chat. We'll collect the proposals, and if it doesn't fit in the next meeting, well, we can see maybe there'll be another slot in April, and probably we'll skip the May meeting, right? Because
I
there's
think
ISMRM,
right,
but yeah, we'll see.
right.
But
Okay. Maybe we'll do an
A-
in-person stuff.
April could be an option.
Right.
Oh yeah, talking of which, it may be a good idea to actually start thinking of a Pulseq party, the ISMRM, or I don't know, Pulseq event. Let's call it an event for now, right?
Definitely. Yeah.
Well, one more
Um,
thing, we will have Roy Ray talk today about his project, but the other slot which we thought about was actually something new, and I've heard that other open source projects, open source communities, they actually do it regularly. They go to the discussions and issues on the site and basically go through the issues and discuss them. And that's something we wanted to try in the second half of the meeting. So it's your chance again. Maybe if you have an issue in mind which you uh, didn't submit yet, maybe you can use the first 20 minutes to briefly, to quickly, post something, and then I will be surprised that there will be some 20 new issues, but we can also then discuss them. And maybe one more thing, I wanted also to use the occasion to make advertisement to an event of which you probably haven't heard of, or maybe did. It is online, I think, from today or yesterday, right, John? So
Yes, it is. This was supposed.
John and myself have put quite a lot of work into preparing it. So we now scheduled and officially posted on the website the new Pulseq sequence programming webinar, virtual meeting, whatever you call it. It's going to be similar to the previous time. Will basically be 3 days in the last week of February. It's Tuesday through Thursday. It will be always actually, so the time which is posted here is not correct, right? So it will be every day from 2 to 5 UTC, right?
Yeah, I think
Yes.
it's 1400
So for
UTC.
1400 to 1700 UTC every day. So basically, we will have 3 hours every day, and that's stretching over 3 days. So it's plenty of possibilities to exchange. Every day we'll have some kind of educational contributions, some kind of user contributions, and also ample time for questions and answers and remarks. So, of course, everybody who is here on this call has have the opportunity to ask questions anyway, but maybe again you can give some answers in the big in the big round, and it's also okay. So, right, that's it from my side. Did I forget something, John?
No, I think that'll do it.
Yeah, I can.
Shall we let
So,
Ray go ahead if there are no other sort of
yeah, I
general
had something.
topics? Yeah, great.
Oh yeah, go ahead.
Hi, this is Anuj from Case Western.
Yeah,
It was nice meeting you, Maxim, at Sedona.
yeah.
Uh,
Hi.
yeah, so I have a small. So I'm working on a sequence, and I think I'm running into a small pulse-seq limitation in how to simultaneously play two gradients during readout. So I can show some pictures, and I can show what I'm doing to go around it. And
Maybe
let's.
let's do it in the second half. I think that's exactly
Yeah,
the question for
sounds good.
for the second part. I think it's we better give right time
Yeah,
to present what he's prepared, and then we can go
yes.
through the questions and answers in the
Yep,
second half. Okay,
But that's a
sounds good.
that sounds great. That's a perfect topic. So that's great.
Thank you.
Let's do it.
okay, very good.
Great. Ray, do you want to go ahead?
So should I share my screen? Let me see.
Should be able to.
That works normally.
Can you
Yes,
see?
it's coming up. Yes.
Okay. Let me see how I can minimize this.
Oh, but you're still not in the presentation mode. At least what we see is a normal PowerPoint. Oh yeah, now it's
Oh,
presentation mode. Okay.
wait. Now,
No, it
sorry.
was fine.
Okay. So now
There seems
everything
to be
fine?
quite a bit of a delay.
Okay. So there's still delay, or now it's fine.
It's still now it's fine. Okay, now it's showing up. But you see, it's about a 30-second delay.
Okay. But can you hear me clearly, or I should do
Yes,
something else?
yes. No,
Okay.
that's absolutely at least. That's fine, but
Okay.
the slides appear with the delays. I have seen that sometimes.
I can hear it as well. It seems okay.
Okay, so thanks a lot for organizing this meeting and inviting me. I'm Rui Tian, and it's my pleasure to present my recent paper about overlap kernel EPI. And this is a paper where the notorious shot-to-shot signal variations in multi-shot EPI meet the graphite and ESPIRI parallel imaging calibration. So the EPI is actually a workhorse for neural imaging. And if you do multi-shot, the reward is clear. It reaches a higher resolution and also less distortion. But it also comes with the curse of the notorious shot-to-shot signal instability that when you combine different shots, you will get artifacts. And here's a example of single-shot EPI in the Pulseq demo folder. And here you can see that you sample a K-space center regions, and the resolution is limited because outside of these regions, everything is empty. And this is because of two reasons. One is that the T2 decay happens along the K-space phase encoding dimension that limits the signal lifetime. And another reason is that the Bezier inhomogeneity also evolves along this axis and makes your image have distortions. So we can definitely make into multiple shots of EPI. There's a case that in Pulseq we implement this multi-shot readout segmented EPI. It's actually similar to the result in Siemens. And the left is the nominal K-space trajectory. Everything looks fine. But actually, when you do this in Pulseq, you probably will have some shifting or shot-to-shot eddy currents. I think we can we close this window? Yeah, you have the shot-to-shot eddy currents that might do some harm to cause shot-to-shot phase inconsistency. And these pictures may be a little bit exaggerating, but at the end, when you scan the image, you may find the actual image details is slightly different than your desired ones because of the shot-to-shot eddy currents. And the more problematic cases comes when you have diffusion weighted imaging indicated by this red block at the beginning of your imaging sequence. And you repeat that for multiple shots. So in these cases, it's a readout, it's a phase interleave multi-shot EPI here, showing on the right corner. We have different shots covered by these different colors of the arrows. So each shot of the image, if you have a intermediate parallel imaging reconstruction, they are weighted by spatially varying phase distributions caused by the diffusion gradients here. And if you simply combine those different shots, you will easily get these multi-shot artifacts that varies across different multi-shot trajectories. So in this case, you have to have this EPI navigator at the back so you can always monitor the K-space by oversampling the K-space center in each shot, but they will cause a lot of additional problems. So our works deal with the basic concept of self-navigation. That is an elegant concept that estimates or correct the shot-to-shot phase inconsistency from the imaging echoes itself. And conventionally, there are two ways to do that. So one is the self-navigated trajectory. That means, like in this propeller trajectory, you repeatedly sample the K-space central regions, but the scan will become a little bit redundant in terms of the sampling efficiency. From the low resolution region that's oversampled in each shot, you can get a low resolution image that has a spatially varying phase fluctuations, which is similarly information provided by the EPI navigator in each shot. And also on the right, we show a self-navigated post-processing or intermediate reconstruction. For example, like the inverse reconstruction, Muse, muscle, Lorax, LLR. There are a lot of different ways to do that. But all of these methods, at the end, they have something similar. So at least a low quality recoverable central regions from each shot should be recoverable. And that means if you consider all the shots combining here, there's not that much substantial parallel imaging acceleration further. So it's a little bit limiting your sampling efficiency if you consider parallel imaging. So we eventually find that there might be a missing scenario for this basic definition of self-navigation. For example, if you have these two shots here, which samples different portions of K-space, and you want to combine these two images at the end, and these are the image representation corresponding to these high-frequency K-space segments. Conventionally, people might assume that there's no self-navigation possible because those are just some edge images of high frequency. However, the K-space information for these sidebands, they are high frequency, but the shot-to-shot phase variation in K-space, which is like a shift invariant kernels, they are low frequency convolved in everywhere. So we proposed we slightly overlap different shots trajectory. So we have these slight, very small overlap regions. We call it shot-to-shot ACS regions. This is a name we inherit from the parallel imaging calibration called auto-calibrating signals. And from this very slight overlap regions, we can apply either east-period or grappa type operation. We treat them as like different RF receiver signals in a very well-defined one step. And then we can obtain these short-to-short phase variations and incorporate that into the final reconstruction that combines different shots. And these will provide the same information as the EPI navigator and can also do the self-navigation in this way with different trunks. So this is where the short-to-short phase fluctuation meets the channel-to-channel receiver sensitivity in guapa and its period. To look closer how this can work, we can do a recap of the conventional calibration in guapa and its period kernels for parallel imaging. So in this conventional case-based data, we define an ACS region for auto calibration. And from these ACS regions, the signals are fully sampled. And basically, we define a neighborhood that a neighborhood of multiple RF receiver data can be multiplied with kernels and then summed to approximate data points within these ACS regions. And that means you can formulate this as a linear equation that a target data as these red dots equals to the source data vector multiplied by these unknown kernels to solve. So if you solve this unknown kernels, this is the information of RF receivers, and you need to be very accurate to be used to have a very robust parallel imaging reconstruction. And the conventional way to do that is each time you shift this interpolation pairs by one K-space index. So in this way, you can get another independent linear equations, but with the same unknowns. By doing this across the entire ACS region, you can have a lot of target data and source data so that you can estimate the known kernels. Because there are a lot of like thousands of shifts, for example, so that means your estimated kernels is very robust because it's a mean estimation from a lot of averaging data. But there are two scenarios that is a little bit overlooked in the past cases. They are trivial. One is that every time we don't need to just sample just right shift the K-space index by one K-space pixels, but we can shift it by many K-space indexes, like this large distance shift. And you will, at exactly the same way, it will provide you with linear, independent linear equations. And there's another trivial cases that we don't need to define this ACS region at the center, but we can define it in the peripheral K-space. These two cases are very trivial. That is because in parallel imaging, if you define these things in the K-space center and shift it one pixel at a time, you will have the highest SNR for estimation accuracy. But in our short-to-short phase fluctuations, this is not really important. So our proposed overlap kernel EPI is actually an intersection of many MRI techniques. So if you have a big picture here, at the end, our goal is to estimate explicitly these short-to-short phase fluctuations. So on the left corner, there was a phaseless encoding MRI I work on for multishot EPI, and where I estimate short interband phase calibration from the non-central K-space overlaps here so that you can correct some interband artifacts. But at that time, I assumed that the phase was constant across the image space. And also in parallel imaging that explicitly extract RF receivers information, people can very robustly calibrate these spatial encoding maps due to the RF receivers. And those are mathematically equivalent to these shot-to-shot phase fluctuation maps, but just along different dimensions. And in the long terms of the low rank matrix completion with like a applied mathematics direction, people have heavily utilized these case-based shift invariant filters. Also, in some recent work where we apply this nonlinear gradient modulation for image acceleration of MRI, we realize at the end we can also auto-calibrate a continuous nonlinear Bezier modulation kernels from the data in a data-driven manner. And that's like a Bezier version of the guapa. And it's much more aggressive Bezier fluctuation compared to this shot-to-shot fluctuation. So if you are familiar with all these techniques, then our solution of this overlap kernel EPI will become very apparent. We can also look at it in another view that for parallel imaging, there are usually two approaches to resolve the receiver channel information. One is explicit kernel or map extraction, that is the graph or is period. And people can also reconstruct the multi-channel data by low-rank matrix completion, like the Saki or low-rex. And when the Saki and low-rex evolved to manage the multi-shot diffusion EPI with low-rank matrix completion approach, that is called muscle. So the question is there a counterpart of the guapa and is periods that can handle the multi-shot diffusion EPI? And this is exactly our technique that obtains the kernel information from the shot-to-shot overlaps, but this time at arbitrary case-based location. And please note that we are not advocating a specific sequence, but it's a general basic manner of doing these things so that this method could become compatible to help a lot of different multiple trajectories to be insensitive to shot-to-shot phase variations. And here are two short mosaic and Pulseq mosaic sequences. There are some people doing similar things, but we realized that by having a very small case-based overlap at the centers, like ACS here, you can very well correct the shot-to-shot eddy current and phase fluctuations in very high quality. And also these two readout segmented and phase interleave EPI are more commercialized nowadays in Siemens and GE and other companies. And here we found that the readout segmented can also be self-navigated by having these ACS regions at these interband regions, which is not at a case-based center. And also for phase interleave, it's a little bit special. It's just like Muse. From each shot of reconstruction, you use this parallel imaging reconstruction to yield a central region that is sort of recoverable, like the ACS. And then we, from this shot-to-shot, from the shot-dependent intermediate reconstruction, we establish this ACS region at a case-based center. In the end, we estimate shot-to-shot phase fluctuation, either kernels or maps, as you like. So this idea is not really new, because, for example, back to 2,000 years ago, this was a famous battle of Red Cliff in ancient China. And those are just a story, like there are two groups that fighting each other. It was a big war. And one group, because their soldiers were coming from the north and they were not familiar with the battle on the water, so they interlock massive warships to become very stable on the water. But shit happens in one evening because the warships are interlocked, so they are too stable. And at the end, they cannot escape from the fires, and everything got burned out. So if you are wondering whether interlocking these so many trunks will work 2,000 years, well, I suggest that you can try this Pulseq demo from Zenodo on your own. And we hope to provide a shorter version in the Pulseq demo folder in the future. And if you have any suggestions of what function you want to keep and what function you want to discard, please don't hesitate to let us know or send us an email afterwards. So if you download this complete set of sequence and reconstruction scripts and also the demo data from the Zenodo, and you will find that they are complete for 4 different multi-shot EPI, and also there's a single shot reference sequence. So these are the sequences, and these are the reconstruction scripts. And for the sequences, please enter your own scan protocols. For example, if you want an imaging or 1D navigator correction scans, what's your pixel size, stroke number, and so on. And once you got the data from the scanner, please enter the sequence of recon parameters in the reconstruction script shown on the left. And so the reconstruction can go through all these functions like grading or panel imaging, reconstruction, distortion. And this is our shot-to-shot phase corrections, and also partial Fourier and so on. So take this as examples from this Zenodo package for this readout segmented self-navigated API. First, you need to generate the sequences by click run of these Pulseq the MATLAB files while setting the variables to be 0 or 1 so that you can either get a readout segmented API sequences or a corresponding 1D navigator scan, which we haven't integrated together. And next, you can generate some graphite spirit ACS scans or the Bezier mapping sequences for distortion corrections on your own. And you can also integrate them on your own afterwards. And after you perform the scan, you save the tweaks or the Pulseq files. Be careful, you need to modify the ID or name specified in our reconstruction scripts. And then you just click run. So now this reconstruction scripts took like about 5 minutes in my standard workstation, and it definitely can be speed up in many different ways. And at this point, you may wonder what would be the optimized multi-shot sequences. And the answer is that there's not a single answer because the optimal solution depends on many, many factors. For the details, you can read my papers. But we can provide a rule of thumb for establishing the shot-to-shot ACS regions. That is, you need to establish a regions that is try to make it close to the K-space center for high SNR. For example, in this phase interleave API, where for different shot of scans, you construct intermediate ACS regions. And these regions can be at the center or can be regions at the different peripheral spaces of K-space, like in muscle or regional muscle at least. But at the K-space centers, you definitely will have the higher SNR. And when the B value increases, these low SNR values, you basically cannot do that much kernel operation there because the SNR is too low. For this readout segmented API, these two bands at the center, like the overlaps, they have higher SNR than these two side bands overlap at the two sides of the K-space with lower SNR. And the overlap region for the left case is about like 50 times 50. And the one on the right is about 200 times 18 pixels. And for the left picture, it was also somehow explored in this technique that combines the muscle with muse followed. But we eventually unified these two steps with what we call the muse with one-step kernel extraction. So this picture just shows you what we are advocating is a basic method for these short-shot corrections to give you more possibility rather than advertising a specific sequences. And so at the end, I want to thank my colleagues at Tübingen. And we are also interested in a lot of further developments for high performance gradients in different sites, like the Sigma X or connectome or impulse gradients. Especially because if you look at these two Pulseq trunks, that is not that commonly used today. They are center out, so they have very short TE. And that means they have very high SNR, which is very helpful for high B-value scans. They might not be that optimized in terms of the distortion levels, but we don't know if this high performance gradient can somehow reduce the distortion problems for those sequences, and then you would have much higher SNR for high B-value scans. And at the end, the special thanks for my past colleagues from Zurich, especially like Frank Hanno and Klaus Bußmann. I developed this phases encoding method for multi-shot API. This is how I enter these areas. And although they are not my co-author anymore, but the discussion substantially inspires this paper of overlap kernel API. And thank you for your attention. If you have any question, just either send me an email or ask after my talk. Thank you.
Great.
Oh, so, yeah. Shall
So
I
much.
stay on my presentation for some time, or I can also stop
Well,
sharing?
it depends
Proceed
on questions. Oh, maybe, yeah. Maybe if you stop share, we'll see more faces. But thank you.
Yeah.
I have one. I just maybe I missed it. But I sort of missed the connection between I get that you're calibrating the graph kernels, not in the center anymore, and that has certain SNR implications. But what is stabilizing? Like, what was the connection with eSpirit, and how is this all solved in one large reconstruction problem, and the overlap regions are somehow used to ensure consistency, or what's the? I missed sort of the central idea.
Maybe I can share my screen again. Maybe it's better. So by the way, which connection you are not very sure about it?
Well, like just the connection with eSpirit and how the sense
Yes,
maps.
okay, I got it. So the connection is like maybe I can put these windows here. Wait for a second. Sorry, I didn't explain this in details because it was supposed to be within 17 minutes. So the connection is that here there are overlaps between different shots of trajectory, and different trajectory, they contains a spatially varying shot-to-shot phase fluctuations. And this shot-to-shot phase fluctuations is just like the receiver-to-receiver RF sensitivity distribution. So in eSpirit or Guapa, you need to explicitly extract this RF receiver, either kernels or maps, from the ACS regions. So in these cases, we define these shot-to-shot overlaps as the so-called ACS. And we mimic that these case-based regions from different shot are just like the case-based regions from different RF receivers. So
Okay.
if the shot-to-shot has no phase fluctuation or amplitude fluctuation, those regions from different shots should be exact the same value. But at the end, they are not. So that means these shot-to-shot regions has been corrupted by convolution of a shot-to-shot phase kernels, just like the Guapa kernel convolve everywhere in the K-space. And so we are exactly using the Guapa and eSpirit type operator. You just swap the dimension of the algorithm. So the algorithm will think of the shot-to-shot dimension as the receiver dimension to extract this shot-to-shot information in one step and use that for the final reconstruction. So actually the computation is much easier compared to eSpirit and Guapa because now we only have two virtual receiver coils. And the calibration region is usually very small. And this doesn't do anything about the reconstruction for the final image reconstruction, because at the end, with all different kinds of methods like Navigator or something like that, you always need to have this shot-to-shot phase inconsistency information that is somehow loaded into the reconstruction. So the final reconstruction, the pipeline is not changing at all. It's just the way we get this information of the shot-to-shot phase fluctuation. The way we get this is new in this paper.
Got it. Great. Okay. Thank you.
Can you explain how this works in the phase interleave case where there is no overlap?
Yes, exactly. So this is the phase interleave cases. So usually, for example, in Muse, for each shot of the acquisition, like the red color like this, people would apply a intermediate parallel imaging reconstruction so that you can sort of get an image, but it's very low quality, but you can still have the rough estimation of the spatial varying phase. And then people, for example, in the original Muse, people use a total variation operation, like a filter, to extract this image space phase variation. So our method differs with the conventional Muse with these post-processing steps. So by this, from the intermediate parallel imaging reconstruction of one shot of acquisition, we go to the K-space and we probe a central 50 times 50 region as the ACS regions, and we apply this kernel operation to extract the shot-to-shot face kernels or shot-to-shot face maps. And the difference is that our kernels is estimated from an averaging of like thousands of linear equations. While if you apply a image space total variations operation, like we show in the paper, if you have some errors in the image space, that's a little bit difficult to get rid of it because there's not that much averaging as we can do this in K-space.
Exactly what Lorex does, right? Does it work better than
yes.
Lorex?
Yes. So the Lorex is like they established calibration region everywhere in K-space, but we are doing something similar to, I think it's this citation here, this muscle and muse. So this is like they first do this Lorex in the K-space center, a lot of Lorex and doing this iteratively, and then they do another muse operation like a Hamming filter in their open source code to get the face maps. So our cases just unify their two steps into one. So you don't need to do this iteratively with this Lorex. So with a central 50 times 50 regions, you can extract this face map from these regions in one step. And you don't need to care anything about these low SNR regions in the peripheral K-space. But in the original Lorex, maybe they have to deal with these space inconsistencies because in their cases, they are locally correcting the K-space inconsistency. But we assume this K-space filters is the same everywhere. So the best place to get the face information is the center.
Does it work better than Lorex?
Okay, well,
the Lorex we haven't tried yet, but the computation should be very.
I think we need to give other people a chance to discuss.
Yeah, we can talk offline. That's okay.
Oh yeah, sorry, yeah, definitely.
Hi, Ryan, may I ask a question?
Maybe Frank.
Hello. Yes. Yes.
Yeah.
Yes. Hello, Rai. So at first, sorry to everybody. I'm joining because of interest in Rai's work. I'm not a Pulseq expert at all. But Rai, I can more or less understand how it works with readout segmenting when you have ACS lines measured at the same time. But in a mosaic manner, like the first picture on the left, you have the auto-calibration lines measured at very different delay from the excitation. And so a little drift of the reference frequency will produce huge changes in phase. And is there no danger that you can misinterpret that for?
Well, I think. May I chime in? The mosaic is center out, so the time is actually not so different.
Okay.
Yeah, sorry, I think it blocks. So the mosaic is center out. So each time, so the upper trunks it starts from somewhere here, like the red dot here, and the blue trunks start from somewhere here, and the ACS regions, they are obtained after an intermediate parallel imaging reconstruction, but only for this central very small segment. So for each shot, you need additional about 2 to 3 lines. But after parallel imaging reconstruction, you can reconstruct a overlap regions that overlaps about like 12 or 15, something like that. That is sufficient for this calibration. It's very high SNR. So their time is not that much different actually. But if you do that not in the central out way, then you may have some problems. And similar to this 4-shot Mosaic, this is a little bit different than this classic sampling scheme in the past days. And also in this readout segmented, I think the neighboring adjacent bands, they are the readout direction, they are flipped. So when those different shots, they arrive in this overlap region between shots, they are sort of like at a similar echo time.
Okay,
Did
thanks.
I address the question? Okay.
Anush has a question.
Yeah, I had a quick one. Yeah. So I worked on like readout segmented EPI several years ago. I implemented it on Canon, and for face correction, we used what Samantha Holdsworth described in a paper using a 2D navigator acquired right after the imaging readout in each shot. And it was still difficult to get face correction right. Like even with physiological motion in the brain, you will see high differences across the shots, and getting a good correction going was hard. So your work is really useful, really beneficial. So congrats on that, and thanks for the talk.
Thank you.
I think the
Thank you so
disconnect, yeah, the disconnect
Thanks
I
so much.
have here is, the disconnect I have here is, so when you draw parallels with Grappa, so Grappa acquires the same ACS lines, which are sampled, shared across all the coils. But in your case, each segment, like the first two segments, have a strip in case space that's shared. Then there's other strip that second and third shot share. So the key space regions across your virtual coils are different. So I might be missing something in your formulation, but how do you
You
account
mean
for
this
that?
ACS region in readout segment?
Yeah, like the orange, the green, the purple, they are at different regions in case space for your different virtual coils.
No, because I think I draw in this way. So because there's overlap, so these arrows cannot, if you are completely overlap, you might not see. So a little bit shifted. But they are actually the same case space location.
Right. The question was that, you know, in your formulation, you now have the overlap is not overlap everything with everything. Like in
Right.
Right. So you can have pairwise equations for your two segments, but not from one segment, one overlap area. Don't extract equations for linking all of your virtual coils if you want for your grapper, right?
You mean this readout segment,
Yeah.
for example, this
Yes.
overlap
So
region
for
is
example,
not,
this orange
not,
only links red coil to the coil, red signals
exactly.
to the yellow signals,
Yes,
and
exactly.
not the others, right? So you have
yes, not exactly. But you need to align the so for each
Right.
interpolate, so for each shot-to-shot ACS region, you can get a pair of two phase map, for example, and then you can get a lot of pairs of two phase maps. But at the end, you need to somehow align them together.
So
because if you
basically
don't align together, it will be a random phase offset. But because
your
like
coils.
because
for
example, the yellow trunks, they have the overlap left and right, but the yellow trunks, they only have one phase, the absolute phase. So that means the phase for the yellow trunk should be the same, no matter if you compare the one from the left or the one comparing the right. So that means at the end, before the shot-to-shot combination, we need to do an alignment between different pairs. If they are not, like the phase interleave, you don't need to do that because everything is overlap at the same regions. Yeah. But also maybe this is, there's also some question like this that's linking to Birking's previous question about how this compared to low ranks. So I think this many people, one person did this similarly a long time ago and cited his paper, is that in this readout segmented EPI, if you look at this very peripheral inter-shot overlaps, for example, in 7-shot readout segmented, and that's very high frequency signals and low SNR. If the SNR p-value is above like 1,500, it's very, very low. And if you do kernel operation, even at this outer shot-to-shot ACS region, it's not the most outer ACS region. The SNR is already low enough to give you some errors in the shot-to-shot kernel operation. And then you'll see a slight blurring of these problems. And these problems, I guess, I haven't tried that, but I guess if you do low racks along these most outer case-based regions, it will be even worse because the SNR doesn't support you to do any estimation. So that is also one reason that in the phase interleave, we implement our method but only look at a central case-based region, like 50 times 50. If you enlarge these regions, we didn't empirically find any advantage by increasing it to 70 times 70. I think the result is very similar. But if you increase to everywhere, like incorporating the most outer ring of the case space, I think it will induce some data that's not really useful. So this is still a theoretical guess.
Okay. I think it's okay. I think it's more or less clear. Do we have more questions? I think it's interesting to give people chances.
I only can see a list
ask.
of 6 pictures here.
Well, I mean, if you stop sharing, maybe we'll see more faces again.
No problem. If anybody has any questions, they can ask later or send me an email afterwards.
Yeah, I think it's a good idea. think it's also probably good that for us to move a little bit on. So thanks Roy again for the very exciting work and nice presentation. Also, thanks for sharing the code. Right.
Thank you. Thanks for the Pulseq for letting me finishing a ISMN abstract within 3 months.
Yeah. Let's see. Should
Okay.
we let Anush maybe ask his question now?
Yeah,
technical one, or
maybe it's good. Let's look at that
Oh,
one.
yeah. Cool. Yeah. Thank you. Yeah. Let me share my sequence diagram. That's
Yep.
easiest to. All right. Can you see the Pulseq sequence diagram? All right.
It's coming. Yes. Okay.
Okay. All right. So I am implementing an expense sequence, which is a spatio-temporal encoding sequence. And I'll give you a quick overview, and then I'll talk about probably a limitation, but may not be. You guys are more of an expert. So there's basically a hard excitation pulse followed by 2 frequency swept 180 pulses. And along with each of the frequency swept pulses, I want to play a GY and GZ gradient. And the other gradients are basically crushers for the 180. And the interesting part is during my readout, along my GX, I want to do an EPI like bipolar readout. And simultaneously, I want to play a flat GZ gradient. Now, this is where I think the constraint comes in. So right now, the GZ is flipped, which is a bypass for me. So ideally, I would want to play a flat GZ because physically they have the same effect, but blipping it up and down is just inefficient. So I would like to play a flat GZ simultaneously with this bipolar. So now I think I can do this in IDEA, because in IDEA, when we add an event, we specify the time at which that event should be played. So I can tell it wherever to play in the block. But in Pulseq, I think as soon as you do add, like seek.add block, time moves forward, right? So imagine that I'm doing this in Pulseq and I add this first lobe here, first readout lobe, and I do seek.add block. As soon as I do that, time has moved forward to the end of this gradient. And now I can't add a flat GZ that is starting from the first gradient. I've tried several shortcuts. I've tried like adding a negative delay. So what I would do is I would add all my readout bipolar gradients and do a negative delay on a flat GZ, but then that doesn't work well. It looks okay, but it doesn't work well. I've also tried the add gradients function. There's a function called add gradients, which allows you to stitch gradients together to create a giant bipolar readout and then add that simultaneously with a flat GZ. But I can't quite remember what was the issue, but that didn't work. I think that didn't work during simulation, but I can't quite remember why it didn't work. So just an open question. Is that possible? Like, is there another way that I'm missing?
Yes. So I have two things for you. So one is, yes, it's certainly possible, because what you can do, so we have this concept of blocks, and one block follows another and they touch each other always. There's no gaps between the blocks, there's no delays or whatever. So what we have is the possibility is to have the gradient be non-zero at the edge of the block. So what you can do theoretically, you can ramp up your gradient or even if you have, imagine you would like to have a gradient which would stretch all over your sequence in some kind of totally asynchronous way. What you can do, you can create this gradient, then you have to cut it into pieces which will be equal to your future block durations. And then when you create your sequence, you have to add these pieces of your gradient to each block, and this will recreate the original gradient from the bits and pieces. So we call it gradient surgery techniques, and we use that, for instance, in the I believe quite a lot in the TSC and in the trophy sequence when we cut through different parts. In your particular case, it's actually even simpler. So what you can do, you can manually create a gradient, which is a ramp up. and it stops after the ramp once you reach your nominal gradient strength. And then for all other blocks, you can create, you can use this extended trapezoid gradient, which can start at any value and end at any value. And a special case of that is that the gradient which starts at a constant value and ends at exactly the same value and only has the flat part, right?
Okay.
So you can have the kinds of square gradient, and then you can put the square gradient into each of the block. So, and that will basically create a continuous gradient, which will not stop anywhere. And then in the end, after you're done with the trajectory, you ramp it down to 0 again, or continue maybe to the spoiler if you need a spoiler. Does it
So
make sense?
basically, yeah, you can basically create these gradient segments with infinite slew, basically flat, and then just add them to each of these bipolar readouts to play them simultaneously. And you said the surgery examples are in DSC and Trufi, so I can look there how to cut the gradients. Right?
Yeah, so
All right.
another, I think so, Semi Laser also has a quite advanced gradient surgery, so you can see how it's done. But
Simulator.
like I said, your example is so much simpler, so you can create the gradient which you need, just because you know what you're doing. And another point I wanted to mention that it is slightly different sequence, but a student from our lab, PhD student who is also co-supervised by me, Andreas Holt, just published a paper with the open source span implementation. So it's called OpenSpan. And
Okay.
I wonder if it's already available at the early view or not. I'm looking for the recent publications. So if I find the link, I will post it into the chat.
Thank you. Appreciate it.
Yeah. Can I just second that point? Like, you know, in the end, you can manually define and alter the various RF and gradient events that go into add block. So the MATLAB or Python toolboxes are useful, but sometimes you still want to, well, generally, I think you still want to check what it actually makes and to see if that's exactly what you want. But once you have the event, it's extremely flexible. Yeah, using the extended trapezoids, extremely flexible. So you can, you know, look at what you have. If it is what you want, then AdBlock will put it in for you. AdBlock has been, as far as I can tell, extremely robust. So see, Maxim, did you wanna? We have 7 minutes to go over open issues in Pulseq.
Yeah, so okay, there was something else we wanted to test drive a little bit, but the time is actually up. Anyway, maybe let's have a look. Basically, I had an idea, but again, maybe we can extend it in the future also to cover the PyPulseq in a way. I'm very much open to kind of see that we cross the link more intensively between MATLAB and Pulseq worlds and PyPulseq worlds. But for now, maybe let's have a look at the Pulseq website and see whether some people can have some answers or comments about what we do with those. So let me share maybe this one. Okay. So here are the issues which I see right now on the web on the Pulseq website, and I just wanted to see that we go through them and see what is there any way we should prioritize it, or maybe any comments, or maybe some people had similar experiences. And also there's like 5 little bit stale pull requests. So once again, we can maybe see how we go about those, because I'm not totally clear myself how we handle that. So I mean, we can maybe pull request a little bit easier. So there was this pull request from Maximilian Graham. If Max is online, maybe you can comment, because I think we now implemented most of it. So I'm a little bit unsure what is the status. Do we do we try to merge it? Is it now solved? Is it supported by the current code in the sufficient extent, or do we still have to do something about it? Okay, it doesn't look like anybody from the team is online. So we'll leave it for the next time. There was one more about X nuclei, where again I didn't have a look at it yet. If the authors are there, maybe that's an occasion. If not, maybe we can also skip that, and I'll need to have a look.
Yes, I'm here.
Oh yeah,
So
okay.
when I wanted to program a potassium sequence, I just had an issue with the conversion, so with the conversion function, because there was just gamma for hydrogen always used, so the 42.
Okay, so the gamma is hard coded in the test report, right?
Yeah, exactly. And in this
Okay.
pull request, I adjusted it to take the gamma defined by the system.
Okay, that's fair enough. Okay, so we should certainly include that. That's a good one. Okay. Okay, thanks.
In PyPulseq, it's fixed already.
Okay, very good. So we'll catch up with that. We have another one where I think it was Matteo, right? Proposing, no, somebody else. Proposing to fix this old case-based calculation function, but actually I was planning to drop and remove it, so that's why I didn't accept it up to now. So I think, is there any desire to use this, or is anybody actually using this non-functional case-based calculation function? Probably no. So I'll probably reject this one. I don't see the reason. And then a long time ago, people were asking to add some more, like adding code spell support or automatic compilation. But at that time, I couldn't verify this thing. So I dropped them. They're probably too far out now. But again, if somebody is willing to revive those pull requests, just maybe write some comments and we'll see what we do. So I think we can have a look at issues then. By the way, we are facing this table undocked issue now and again, and I wonder, so it's Siemens specific, and it happens on the recent Numaris 10, Numaris X systems, where you suddenly get this message box that your table is undocked. That actually happens on both of our scanners, which have fixed tables, so the table cannot be undocked. So it's very clearly I miss information. So if you see this problem, the best thing to do is to watch. So what basically happens is that the control process of the scanner, which controls the execution of the sequences, crashes. The table cannot talk to the control process, and the table is the first item in the whole infrastructure to detect that something is wrong. That's why this message comes. And the best thing in my experience is just wait a minute. You can see the screen if you really can look into the magnet from your working place. You can see the screen or these tablets on the sides of the magnet, or on some other models it's on the top of the magnet, which where you see that it goes black and then there's some kind of booting sequence showing up. After a minute or so, all our scanners recover. They show again the normal, table control or whatever coil selection interface. And from that moment on, the scanner is usable again, actually works without any issues. If you start at this point to try to restart something or do something with the scanner to trying to fix the problem manually, in my experience, it actually gets worse and you end up having to restart the whole scanner completely. So somehow the best strategy, if you see this table and dock problem, is to not to do nothing or to do exactly nothing for a minute or so until this scanner control process restarts. We actually don't know what is happening. We had some interactions. We found some situation when it was happening, but this one is fixed already, and for now we really have no idea how to fix the rest. What happens is that this process crashes without any log entry or whatever. So it happens very sporadically. It happens quite rarely after the recent fixes, but it still happens, and we don't know what to do. So if it starts to happen annoyingly often with your particular sequence, it's actually good news because maybe we can use it to debug the situation for everybody. So if you get this very often, please let us know, and we'll be happy to try to look at it and try to fix it.
There was just a quick message in the chat here if you want to look at that.
Okay, so Max is a CMR, and the table undocked, Marta confirmed that it was exactly a similar experience. Yeah, so basically what we assume is that what happens is that there's some kind of, so for the situation where we could reproduce the problem was related to using too many shaped gradient in the sequence. And so once you overload the shape memory of the scanner, that case it was some kind of a relatively crazy 3D spiral sequence. It was crashing the process. So again, it's a Siemens bug. It should have given an error, but it was crashing. And in the new interpreter, which we will hope to release somewhere in February, we now found a way to be much more economical about the way how we use memory, shape memory. So many of the sequences which don't run with the current interpreter will actually start running with the new one. So there is a light in the end of the tunnel, but there are also other problems. So apparently some other situations also produce this. Maybe the shape memory is not properly released, but we don't know. We don't really see any meaningful log entries, and I tried to ask Siemens people several times. Nobody knows what's happening. So this is a little bit unfortunate, but once again, if you've learned to reproduce the problem, preferably with the new interpreter, which you don't have yet, please let us know. So after we release the new interpreter, if you observe this kind of instability, contact us as soon as possible, especially if you find a way to really reproduce the situation sufficiently often. Okay. So
thank you.
Sure. I think we're out of time. So from the issues which we see here, are there any particularly burning ones? We have a situation that actually the compilation doesn't work with this with these flags, and it's totally fine to just comment them out. So if on your system they are not known, then this needs to be just comment out the line which use this additional flags. The problem is a little bit that because of the diversity of the systems, of the Siemens systems in the field, we have no overview around at which time point these flags were introduced, so we cannot really detect the situation for now. So, but in the next version, we'll basically remove it in the standard in the standard version for the older Fundamarius 4 and will not even try to use them. So that's probably the only issue I would be aware of. And I think most of the other issues are somehow answered. The one here is actually an interesting one. Oh, actually, we didn't respond. I didn't respond to that. That's too bad because I didn't. I've seen that, but anyway. So in general, if you want to use API and or any other sequence which requires external grapple lines, we in Pulseq don't yet have a reliable way of implementing it. So try it for yourselves. Try to see whether you can mimic the logic of the flags and switches, basically the labels from by observing how the product sequences work. But for this particular case, I know that Jinping from our group has produced a special version of the Pulseq interpreter, which was able to do that. But we didn't have time to merge it into the main line, so it will probably still remain a little bit of a challenge for the next version. Okay, so I think we probably have to stop now unless there's some burning questions.
Yeah, that sounds good. It's probably good to try to stay to 1 hour or so.
Yes, I think so.
Great. This is great. Thanks a lot for the questions. And I love all this technical stuff. That's the good stuff.
Mm.
Okay. Let's wrap it up. So let's aim for March 6th for the next meeting. I'll send out a notice.
Yes.
So,
And we're looking for volunteers to give a scientific presentation next time, or a volunteer.
great.
Okay.
Okay. Take care, everyone.
Right. Thanks, guys. Bye.
Thank you.
Bye-bye.
Bye.
Thank you.
Bye.
Bye.